Ladies and gentlemen, dear guest of honor, Professor Lisa Feldman Barrett, I welcome you all today to this special event, another highlight of this week of conferences and the last week of our term. I'm speaking to you today as the head of the Cognitive Science Research Hub at the University of Vienna, one of our horizontal structures dedicated to interfaculty, transdisciplinary, and interdisciplinary research. And as such, we study and promote interdisciplinary research in the field of cognitive science with a very broad approach. Yeah, we sometimes say the typical Viennese approach, Marcos, that's how we call it, which ranges from molecules and how they, in animals, promote their ability to think, across animal thinking, a lot of psychology, philosophical, and also studies in the humanities that perhaps could not be studied and answered without the cooperation with empirical sciences. Also, although we are particularly active in basic research, we also see that the current challenges facing our societies can hardly be mastered without a comprehensive understanding of our mental processes. Because our behavior in many ways is due to human thinking, and today's world's most dangerous challenges in politics, democracy, and climate are often based and rooted in our thinking. Well, there are also, now coming closer to what we hear today, or what Lisa Feldman Barrett is the expert for, there are emotions behind everything that people do. They are the overpowering and ingenious drives of life that control our actions. So the task of the brain is to guide our future behavior. Brain and mind must therefore try to predict what will happen. And therefore today's talk concerns a fascinating building block regarding the question of how our thinking and our actions, by anticipating the future, providing the corresponding information that we will need in the future. This fits excellently with our strategy to research future thinking in all its facets in the future. I would like to thank everybody who helped organize today's event, and I hope that you as our guest will be that you as our guests will all be inspired by today's lecture. And of course, I thank you very much for sharing your knowledge with us and the willingness to be here today in Vienna. It's a great, great pleasure. And before you start, I ask Georgia to give a short introduction to your person. Well, in Austria, you know, we could go on now welcoming every person by name with a little applause, and it's a pleasure. It's pleasant, but it takes a lot of time, and I think some of you are keen to hear more about the science today, and therefore I thank you very much for your attention.
So thank you everybody for being here, and especially for Lisa that joined us today. I had already the pleasure to meet her yesterday, and it's a wonderful experience. So I will read few notes that I wrote about, and I got in internet, as many of us Google people, and also a personal note that is, I think is very meaningful also why she's here. So Professor Lisa Feldman Barrett is a university distinguished professor of psychology of Northeastern University in Boston, where she directs the Interdisciplinary Affective Science Laboratory. She is among the top 0.1% most cited scientists in the world for her revolutionary research in psychology and neuroscience, renowned for the pioneeristic work on the brain as an organ of prediction. We will know and we'll hear more today, responsible for constructing our perception, emotion, and bodily state through continuous, contextually informed anticipation and adjustment. So, and I still, a little bit introduction of Siggy yesterday that I liked a lot. So our brain is not primarily meant to read books, hear music, or see movie, or in our case, as we are scientists, to plan sophisticated experiments, but to regulate about internal systems, maintaining homeostasis by predicting and managing the physiological needs. But besides this groundbreaking work on system-level models of brain and body mechanism, I think Lisa is an inspiration to look for the big picture, pushing us to the boundary of human knowledge. And this is, for me, one of the things that I found mind-blowing. So the personal notice that I think many of us decide to become scientists because we are fundamentally curious and driven by big questions. You must have one. This is big in your heart. My personal one is, what is reality? It's really a big one. And actually, in my search, I have mostly looked for an answer, really not in psychology, not in neuroscience, but in physics, quantum physics, sometimes philosophy. But I think Lisa is bold enough to ask this question in the domain of psychology and neuroscience and eventually be able to answer it. So her recent work on relational realism, the idea that everything that is real exists only in relation to something else, force us to reflect on this big question. And if you grasp the power of this idea, it's mind-blowing. So yesterday I just discovered that we have a common favorite author in quantum physics, a theoretical quantum physicist, that is Carlo Rovelli. And I had the chance to see him in Vienna. So I'm really so happy that he is also known to Lisa. And who knows, maybe one day the two of them will do something big. So regarding more the career, Lisa has published almost 300 peer-reviewed scientific papers, among them Science, Nature Neuroscience, and other top journals in psychology, cognitive neuroscience, and bestseller books, as you know, as 7.5 Lessons About the Brain and How Emotions Are Made. She's also very engaged with the public, and she writes regularly for popular press, and she has several podcasts, interview radio programs worldwide, with, for example, her TED Talks being viewed over 7 million times. So now, with no further ado, I'm really very honored to welcome Lisa on the stage.
So now we bring this down to where a short person can. All right, so let me first start by saying it's a pleasure. It's a pleasure to be here. It's a pleasure to have the opportunity to talk to you. And I thank you again for that really lovely introduction. I would love to do something with Carlo Rovelli. I think you should tell him. You should tell him. So let me, before I start, let me ask how many of you were at the talk yesterday? Okay, so I expect you, this is the same talk. So I guess I should apologize a little. There are a couple of few things, little tidbits I've added, but I expect you guys to seriously laugh at all my jokes. You have no excuse, because you can predict when they're coming. Yeah, okay. So this is going to be a high-level conceptual talk, but there will be moments of where we sort of dip our toes into some data. So in that interest of looking at a little bit of brain data, how many of you have no experience looking at brain data? You would need, okay. So what this means is that I'm going to stop occasionally and orient you to what you're seeing. And that means we're going to go just a tiny bit slower than I would at a meeting where people are familiar with what they're looking at. Otherwise, you just have complete experiential blindness to what you're looking at, okay? And I would say the goal here is to outline some big ideas about a broad approach to brain function and how what it reveals to us about how the mind works. And what I'm going to do is I'm going to cover one basic operating principle of the brain, a major hypothesis that's very anatomically inspired about how the brain predicts, and then an implication for what it means for development. This is for you, Georgia. And when I begin, before I begin, I always like to thank the other directors of the lab, Dr. Karen Quigley, who's standing beside me, and Dr. Jordan Theriault, who is joining our lab as a director in the fall. And then also the wonderful students and postdocs and research scientists we've worked with at Northeastern University and Massachusetts General Hospital. And I'm showing you about 3 generations of mentees here, because I think it's really important to understand that I am like the front man here, right? So what I mean by that is we sometimes have this illusion of it's like the great man view of science. It's mostly men, so, you know. But the great person view, like one person has, you know, all the ideas and all the data. And I'm actually the front person for an extremely large group of people who've been collaborating and working together for an extremely long time. And this is just a sample of the people whose work I'm going to talk about today. So I'm going to start off with this very basic question, which is, what is the brain's most important job? And to answer this question, we're going to go back 550 million years, give or take, to a time when the world was ruled by creatures who had no brains. Kind of like now, thank you. Good job. And I say that joke has extra meaning today after yesterday's debate. All I can say is on behalf of all Americans, I apologize. And I wasn't even born an American, so, you know, anyways. But so kind of like now, except back then, creatures actually didn't need brains. And a really good example is this little guy, the Amphioxus. Its body is tiny. It has a very small number of organs, no actual organ systems, and virtually no systems to sense the outside world. And also these little guys have no brain because they don't need a brain. But over evolutionary time, animals' bodies got larger and larger. These larger bodies had more internal visceral systems to coordinate and control, systems to shuttle nutrients like oxygen and glucose, systems to eliminate waste. Also, animals evolved for the first time new sensory organs, eyes, a lateral line system to sense the world around them, which further perturbed those inner systems, which had to be coordinated in a metabolically efficient way. So in short, as bodies became more complicated, they needed a central command center to regulate them and coordinate them efficiently as animals move around in a highly variable and only partly predictable world. And that's really what your brain is. So your brain's most important job is coordinating and efficiently regulating the many, many tissues and organs of your body. So consider that right now, for example, as you listen to this talk, your very own central command center, your brain, is regulating your extremely complicated body. It's supervising over 600 muscles. It's balancing dozens of hormones, which are coursing through your blood at about 4 to 5 liters a minute. At the same time, it's regulating billions of energy cells, including its own, the energy of billions of cells, including its own cells. It's directing other organs as they digest food, excrete waste, and fight illness. And the name for this process is allostasis. How many of you here have heard of allostasis? Oh, so a good number. That's great. Allostasis means energy regulation is efficient at all levels of output. For example, metabolism can be efficient when you're asleep, and it can also be efficient when you go for a morning run. When I go for a morning run, I don't run anymore, but when I did, I can assure you that I was not in allostasis. But the point being that sometimes we throw ourselves out of allostasis. That's what interval training is, right? In order for our brains can learn to get us back in quickly. And in our lab, we focus on allostasis instead of the more familiar homeostasis, which is about regulating back to a set point. So in allostasis, there is no set point. The goal is efficient regulation, metabolically efficient regulation at all levels of output, and the regulation is predictive, whereas in homeostasis, it's reactive. Now, individual systems in an organism can work by homeostasis, like temperature regulation in humans works by homeostasis. But all of the systems together are coordinated, regulated by the broader allostatic efforts of the brain. Allostasis has been defined the core task of all brains by the physiologists Peter Sterling and Simon Laughlin, who wrote in a fabulous book called the Principles of Neural Design. The core task of all brains is to regulate the organism's internal milieu, which means the internal systems of the body, by anticipating the needs of the body and attempting to meet those needs before they arise. And they came to this conclusion on the basis of extensive cross-species analyses of the metabolic costs of electrical signaling, and then derived basic principles of neural design based on that cost. As the brain regulates and coordinates the systems of the body, it's also predictively modeling the sensory state of the body, which is a process called interoception. Many scientists define interoception as awareness of physical sensations, like feeling your heartbeat in your chest. But interoception is really broader than that. It's about the brain's predictive modeling of the sensory changes that result from allostasis. And this supports allostasis in the same way that somatosensory signaling supports skeletal-motor control. Now, your mood, or what scientists like me call affect, is linked to interoception. Interoception is very, very high dimensional. There's a lot going on inside your body, and those, the sensory surfaces inside your body are constantly streaming signals to your brain. We're largely unaware, we're wired essentially to be largely unaware of the, interoceptive drama going on inside our own bodies. If we were aware, we would never pay attention to anything outside our own skin ever. But the brain makes itself aware of the sort of, as a general barometer, makes itself aware of the allostatic conditions of the body as mood or affect, which we can think of as a compressed, lower dimensional summary of that very high dimensional drama going on inside the body. And so this low dimensional summary manifests itself as feelings of pleasure, displeasure, comfort, discomfort, fatigue, activation. So it's a quick and dirty summary of your allostatic status without a lot of detail. And the important distinction I want to make here is that affect is not emotion. Because your brain is always regulating and modeling your body, affect is always with you every moment of your life, whether you are emotional or not. So valence and arousal are properties of consciousness. They are not emotions, they're not processes, they are properties of consciousness, and they are present even when affect is not in the forefront of your awareness. So when you're feeling pretty good, allostasis is probably going pretty well, and when you're feeling like crap, really fatigued or really distressed, allostasis is probably less efficient. It might mean that something is wrong. It also might mean that you're just doing something really hard and you're metabolically compromised in the moment, like you've exercised for a long time, for example. Or it could mean that you're learning something new, which is also metabolically expensive. Or it might mean that you're dealing with persistent uncertainty, which for humans is incredibly metabolically expensive. It could also just mean that you're sleep deprived. So how much of the brain, so let me just back up and say, before I go on to this next bit, as psychologists and neuroscientists, we're used to thinking that when we feel bad, it means something is wrong. And I think that you can look back to the evolution of the brain and come up with a reasonable story for why it might work that way, which I'm happy to talk about later. But the point that I'm making here is that feeling bad doesn't mean something is wrong. Doesn't mean something is wrong in the world, doesn't mean something is wrong with you. It could just mean you're doing something really hard, or that you need to take a nap, or that you need to drink some water, or that you need to get a hug from a friend. It doesn't always mean that there's a problem. Okay, so how much of the brain is devoted to allostasis? We set out to answer this question in our lab a number of years ago using resting state connectivity analyses. So resting state, how many of you don't know what a resting state analysis, like a resting state connectivity analysis is? Okay, well, for you, this is for you. So, so neuroscientists will have participants lie in a scanner, sometimes with their eyes open, sometimes with their eyes closed. They're lying perfectly still, and the brain is not being probed with a stimulus on screen. So the subject is just, their mind is wandering, and you're scanning their brains. That's basically it. It's called the resting state. We're going to talk about the resting state later. But there's a lot of evidence to suggest that when you scan subjects and you look at the correlations between bulbs, the fMRI signal, that this reveals something to you about the connections that are actually the anatomical connections in the brain. So what we did is we used published track tracing studies of macaques that have traced anatomical connections. And we identified the cortical areas, the areas of the cerebral cortex, with direct connections to the brainstem nuclei that regulate the autonomic nervous system, the immune system, the endocrine system. So in other words, we were looking for the cortical areas that are important for performing allostasis. So these are macaque brains, and I don't have them labeled, but this is a lateral view of the brain, of a macaque brain, and this is a medial view. So if you take the two hemispheres and you crack them open like an egg, this is one, you're looking at the sort of like the inside surface of one view, medial view of a macaque brain. And this is the front, and this is the back front. So we identified regions in the cortex that were connected to these subcortical regions that perform allostasis. And then we identified homologous voxels in the human brain, and we used them as seed regions. And then we examined the resting state connectivity between each seed region and the rest of the brain. So basically, we were looking at the time course of signal change in the seed region, and we asked throughout the rest of the brain, where else is there signal fluctuations that correlate with the signal fluctuations in that seed? This is a standard practice for looking at what's called intrinsic connectivity. And we had many, many seed regions, so we had many, many maps. And then what we did is we computed the similarity between those maps, and then we did a cluster analysis to identify the brain system for allostasis. And then we checked these results against another subsample of subjects using standard imaging, totaling, I think that was over 700 subjects in total. And then we replicated the whole thing in a very powerful 7 Tesla imaging study with almost 100 subjects. So we had over 800 subjects, more or less, where we replicated these results. This analysis produced one integrated system that was clustered into 2 very well-known intrinsic networks. So these are networks that have been identified many, many times in the neuroscience literature. And for any of you who are familiar with that literature, what we identified was the default mode network and the salience network. And these networks overlap in hubs which are, in the connectomics literature, thought to be serve as the backbone for communication in the brain. So these hubs are considered to be important for synchronizing brain activity throughout the entire brain. And they are also crucially important in allostasis. And I'm going to get to why it's important that we found these 2 networks in particular in a moment from now. But the point is that together, These two intrinsic networks and where they overlap make up an integrated system for maintaining allostasis. And here's the replication in 7 Tesla in almost 100 subjects. And interestingly, and not surprisingly, this system also includes primary interoceptive cortex, which is modeling the sensory consequences of allostatic regulation. So here I'm going to present the results. It's exactly the same results, but now I'm projecting this into the volume of the brain, and I've labeled the front and back of the brain so you can see. And so one thing to notice is that, first of all, the system is widely distributed throughout the cerebral cortex. So it's not just the subcortical areas of your brain that are important for allostasis. Your cortex is very involved. In addition, there's a whole matrix of subcortical areas which are involved, which is not surprising. This is sort of how we, how we selected our cortical seeds in the first place. But here is what's surprising. This system also includes the hippocampus. Does anyone know what I'm showing you here is it's a slice of the longitudinal axis of the hippocampus. So you're looking at a lateral slice of the hippocampus and its connectivity to the rest of these regions. What is the hippocampus known for? What do scientists think its job is? Memory, or spatial navigation if you study rats. It turns out that the hippocampus has more endocrine receptors than any other structure in your entire brain except the hypothalamus, to which it is directly connected. There is a loop from the hypothalamus to the hippocampus and back. And it turns out that the hippocampus is important for all kinds of allostatic functions. And right now, I'm working with a couple of other scientists one who studies spatial navigation, one who studies olfaction, one who studies episodic memory, and Karen Quigley, who's the co-director of our lab, who's a physiologist. And our hypothesis is that the hippocampus is not constructing spatial maps, it's not constructing social maps, it's constructing allostatically weighted maps. The reason why do animals need to remember how to move around in the world and where to go? Because they have to find food, and they have to avoid predators. And so the hypothesis that we're working with here is that the main function of the hypothalamus is allostatic, and as a consequence, we have these other functions that have emerged. Also, another major structure that's involved in this system is the cerebellum. Here I'm showing you a horizontal slice, so if I slice the brain like this and I pop the top off and we look down, clean off all the blood, that's what you see. What is the cerebellum known for? What do people think is its major function? Fine-tuning motor movements. The cerebellum actually evolved in vertebrates as a sensory structure. And there are many papers that have been published talking about the non-motor movement-related phenomena that involve the cerebellum, and no one knows why. So why am I belaboring this point? Because I think scientists tend to label things in terms of their own interests. If you're interested in memory, you think the hippocampus is a memory structure. If you're interested in spatial navigation, you think the hippocampus is a spatial navigation structure. And then there are lots and lots of papers that get published where people debate which one is actually more primary, or try to figure out how to integrate those two into a theory that will account for both. The cerebellum is another really good example. People puzzle. They puzzle. Why would the cerebellum be involved in thinking? Why would it be involved in memory? And so the point that I'm making here is that some of the big debates that we have, the big puzzles that we have because of how we labeled and categorized structures. And instead, if you just look at the evolution and the anatomy of the brain, you get a bunch of clues. So in the same way that physicists use math as a rational way to discover, new phenomena, in our lab we use anatomy actually and evolutionary biology, and it leads us to not be so puzzled when we see something like this. So pretty much everything that these networks are doing, they're doing while they maintain allostasis. And as you can see here, they're doing many, many things. So this is a summary of data from more than 6,000 published papers from 10 years ago. These findings highlight, they match and they emphasize many other findings since then, which indicate that psychological phenomena don't have dedicated brain parts. There is no emotional system in your brain. There is no cognitive system in your brain. Every psychological phenomenon, every event, is a whole brain event. And when studies are designed properly, this becomes very, very evident. That every thought, every feeling, every movement, every perception can be thought of as a whole brain event. So I'm going to give you a couple of examples of why this hypothesis about allostasis is important and how it changes our understanding of existing findings. Actually, I'm going a little slower, so I might just give you one example, the sort of more important example for today. And that's the following. It involves this midbrain structure called the periaqueductal gray, or the PAG. So how many of you have heard or read that your brain contains innate circuits for fight or flight? Yeah, this is, I'll tell you that in the US, this is a very, very, not only is popular in neuroscience circles, but it's very popular in the media, in the popular press, in coaching and executive coaching and so on. The PAG is supposed to contain fight or flight circuits that are activated during predatory threat. Now the PAG is a very small cylinder that surrounds the cerebral aqueduct, so it's extremely difficult to see using standard 3 Tesla brain imaging. And we completed a study in which we scanned people using ultra-high resolution 7 Tesla scanning to examine whether it's more justified to think of the periaqueductal gray as just regulating the autonomic nervous system all the time, not just during threat. And so while we were scanning subjects, we gave them an NBAC task, a working memory task. How many of you know what an NBAC task is? Okay, so I'm just going to remind you how absolutely, utterly trivial this task is. It's easier than remembering a phone number, especially once you get past a certain age. So we show subjects, on a given trial, we show subjects a letter, okay? Then the next trial, we show them another letter, and we say, was this the same or different than the last trial? So was it the same or different? Okay, and now, the same or different than the last trial? Okay, that's the task. This task changed the magnitude of bold signal. When I say bold signal, that's just the fMRI signal in the PAG. And notably, it also changed subjects' heart rate and their respiratory rate. So to see this, I want to show you this because I think it's a beautiful example. The work that I'm going to show you is done by Jordan Terrio, who's not here today, but he was at the FENS meeting. He led this work. So what we did is we took this cylinder and we divided it up into 7 cross-sectional segments. So here's one cross-sectional segment, if my computer will cooperate. There we go. So there's one cross-sectional segment. So just to understand what you're looking at, the PAG would, this is a coronal segment. So if you cut the brain this way and you pop the top off, that's what you're looking at. And so the PAG really is a tube that comes in out of and then into the screen. So that's the structure. It's a very, very small structure. It's very, very hard to image. And what we did is we took one, so this is an example of one cross-section, and then what we did is we peeled it like an onion with radial strips from the inside, near the cerebral aqueduct, which is in black, to the outside. And then we laid each one flat. So what that gave us was an entire flat map of the entire PAG, voxel by voxel, that we could look at. And so that's what we did here. We laid out all of the strips into one flat map where each dot is a voxel. The x-axis illustrates the position of the voxel along the rostral caudal axis. So from the top of that column to the cylinder to the bottom. And then the x-axis represents the voxel's position relative to the aqueduct, the black center, to the outer rim. So on the left is the innermost voxels, and on the right is the outermost voxels. And the red represents an increase in the bold signal relative to baseline, and a blue dot indicates a decrease, significant decrease. And so what this shows is that in a task, which is easier than remembering a phone number, you have modulation of the PAG. When we have subjects remember 3 back instead of 1 back, we see a little bit more intense changes which in the PAG signal, which is what we expected to see. But findings like this are consistent. They are inconsistent with the idea that the PAG contains, that those voxels are part of circuits for fight and flight. So again, when you are faced with a threat, your PAG will work very hard. But that doesn't mean that the circuits are for fight and flight. They are for regulating your autonomic nervous system, which happens to be very intensely modulated when you are threatened. And I think that it's time for those of us who write about this and think about this to stop claiming that there are innate fight and flight circuits on the basis of evidence where studies were designed to get big effects, right? So what did scientists do in the 1940s and 50s and 60s? What do they do now? They design studies that will modulate the PAG intensely, and then they label the function of the PAG in line with their experimental manipulations. It's a problem. It's a pervasive problem. I'm going to skip the second implication because I think it's about resting state, and I don't know that it's so important here. And I want to move us along because I want to leave enough time for questions. So I'll just say that taken together, so far what I've showed you suggests that the brain's regulation of the body to which it is attached is the brain and the body are always communicating. They're like two systems which are constantly communicating with each other. And your brain's regulation of your body is at the core of brain structure and at the core of brain function, and therefore at the core of your mind. It doesn't matter whether you're experiencing a thought or a feeling or a perception. At the same time, your brain is also constantly regulating your body and modeling the sensory changes that are anticipated. So your brain's most important job is not thinking. Can I tell you, this got me into so much shit with some cognitive scientists who I know. They were really pissed at me when I wrote this, but I stand by it. Your brain's most important job is not thinking, it's not feeling, it's not seeing or hearing or sensing in any way. Your brain, of course, your brain does all those things, but that's not the brain's most important job. The brain's most important job is regulating your very, very, very complicated body. And until now, the brain's regulation of the body has largely been ignored from the neuroscience of the mind I think because largely we're unaware of all the drama going on inside our own bodies. And frankly, the dominant paradigm in psychology is to start, and neuroscience is to start with our own experience and then go looking for the physical basis of that experience in the brain of humans and other animals. And so what that's led to is a major blind spot in literature that I think can be corrected. One important implication here is that if we really want to understand basic psychological phenomena, like how does a brain plan action? How does a brain decide? What is mood? We actually have to know something about how the brain is managing allostasis in those moments. And another implication I think here is that the distinctions between mental and physical, cognitive and emotional, were created by human brains, right? Not discovered. We didn't discover those distinctions. We've made them, and maybe they're not so useful anymore. So the next, we're now going to move to a hypothesis that is related to this allostatic principle. And so we're going to talk about where allostasis might be happening. No, sorry, we're going to move from where allostasis might be happening to how the brain might be managing, achieving allostasis. And you'll remember that allostasis is defined as the predictive regulation of the body. And it turns out the most efficient way to regulate any system is to predict its needs in advance, predict its state in advance, and then adjust and correct those predictions when necessary. Now, in neuroscience, and to some extent in psychology, in philosophy, in a broad swath of fields, there's a growing research tradition that's emerged in the last decade. Actually, its roots are much, much older than the last decade, but I think the bulk of papers have really been published in the last maybe decade, maybe 15 years, focusing on understanding the predictive dynamics of the brain. There are literally hundreds of published papers with evidence now showing that the brain functions predictively. And in the interest of time, what I'm going to do is I'm just going to provide you with a gist, description of how the brain accomplishes prediction, and I'm going to put our own lab's theoretical twist on this, our own spin on this, this hypothesis. So using a cartoon example, the general idea is that your actions and your experiences begin as memories that are assembled to, they're assembled to be similar to the present in some way. You're not aware of yourself remembering anything, but what your brain is doing is re-implementing signal ensembles from the past that are similar to the present in some way as a way of predicting the immediate future. In psychology, a group of things which are similar to one another is a category. So prediction signals are momentary categories, or what Larry Barcelow would call an ad hoc category, and your brain is a category constructor. A category is a grouping of possible futures that are continually tested against the sensory signals that are constantly streaming to the brain from the sensory surfaces of your body. You might even say that those incoming sensory signals from the sensory surfaces, you have sensory surfaces, you know, in your retinas are sensory surfaces, your cochlea sensory, but you also have lots of sensory surfaces inside your body, constantly sending signals. You might even say that those signals are helping to select the specific prediction signals from the available category that will guide action and become your experience. So we think about the brain as controlling the body, but in a very real sense, we can also think about the signals from the body as constraining the brain. Where the brain, you know, in the associative trajectory of the brain, what's possible in the next moment is being somewhat constrained by the sensory signals from the sensory surfaces. And when we end up talking, if we have time to talk about relational realism, I think this is an important, this is actually an important aspect that sometimes gets overlooked. So if you think about the last time that you were thirsty and you drank a glass of water, within seconds of draining the last drops, you probably felt less thirsty. But the really interesting thing is that it takes about 20 minutes for water to change the osmolarity of your blood and for the corresponding signals to reach your brain. So that means that your thirst is being relieved 20 minutes too early. Because over your lifetime, your brain has had thousands and thousands of opportunities to learn that certain sensory and motor patterns in one moment, which are involved in drinking, result in the quenching of thirst in another moment. And so your brain predicts and begins to construct that experience before the sensory signals arise. So here is an example that I'm going to give you that offers two more interesting insights about prediction signals. How many of you have seen this video before, other than the 4 people who were at the, okay. So first, for the rest of you, I'll bet a lot of money, actually, that you've never actually seen electrical towers playing jump rope before in your life. But you can, I hope you can understand that's what's happening in this image, because your brain was able to combine bits and pieces of past experiences to make a completely novel prediction. In cognitive science, this is called conceptual combination. And it's a very, very powerful capacity of the brain. But this example also demonstrates something, I think, really important about the embodied nature of prediction signals. How many of you can feel the tower, feel the vibration of the tower hitting the ground as a thud in your chest? And try harder. And how many of you can actually hear the sound? Yeah, I can hear it too. If so, then your brain is changing the firing of its own sensory neurons in the absence of the relevant sensory input, only on the basis of visual input. This is an example of what your brain does every time it creates a prediction. Prediction signals are your brain literally changing the firing of its own sensory neurons to anticipate sensory signals, and in the process begins to construct a conscious experience before the signals actually arise. How many of you have ever had a song going through your head that you can't get rid of, right? And this is what, I mean, I did this at the FENS meeting, I just thought of it. How many of you are Queen fans? You want me to put a song in your head? Right? I did a video, I put this on a video actually. Right? Okay. All right. And here's another example. I know I'm going a little slow, but this stuff is like it's just too much fun. This is an example I recently found on TikTok. But I think it's so awesome that I just wanted to share it with you. So this is a traffic light. What colors do you see? Shout it out. Great. Okay. Here's a really cool thing. Now what colors do you see? Who said gray? Who said gray? You, my friend. That's interesting. Most people see the top light as red. Who sees it as red? Okay. The interesting thing is that this is a cyan filter. And your experience of red, except for you, because you're experiencing gray, your experience of red is completely remembered. It's completely simulated. It's completely a prediction. And I know this because cyan filters block all wavelengths of light that neurotypical humans see as red. And I'll show you that that's the case. So what this tells us is that every experience you have is some combination of the remembered past, your predictions, formed as categories, and the sensory present. Okay, here's the last example for today. I know. I feel like it would be very fun to teach an entire course in psychology just with illusions. Okay, so what happens when your brain can't construct a prediction signal? So this is a Mooney image, and right now, for most people, their brain is searching through a lifetime of past experience, issuing thousands of guesses, weighing probabilities, trying to answer the question, what is this most like? Not what is this, but what is this most like? Your brain's trying to construct a category of prediction signals. A fighting warrior. Anybody else? Shout out what you see. Octopus? Some people see a bunny. Someone saw a lobster. A hungry person. Saw a lobster. Who's still seeing black and white blobs? Okay. If that's the case, then your brain hasn't been able to construct a good category of predictions, and so you are in a state called experiential blindness. And now I'm going to cure you of your blindness. I'll leave my evangelical, yeah. Are you, it's so hard to have, it's just even 4 of you here because it doesn't, I know you've seen me do this already. So what I said yesterday, which is actually absolutely true, is that, in the United States, there's a very healthy evangelical community. And so when I'm on stage, I like to get up in front of people and say, do you want me to cure your blindness? Who wants to have their blindness cured? Yeah. And then I usually make them do it louder. So I'm going to cure your blindness right now. That is the most gratifying sound when audiences do that. Like, oh. So you can see it's a photograph of a bee. And the bee has antenna, and it has a head. It's got a fuzzy body and a fuzzy little bum. It's got shiny wings. And now who sees a bee or a part of a bee? Yeah, most people do. And the reason why is interesting, right? Because this image has not changed from the first time you saw it. What's changed is you. Your brain has changed. Your brain is now able to construct a set of predictions that it wasn't able to construct before I showed you that color photograph. And as those prediction signals are changing your experience of these blobs right now. So your brain is constructing an image of a bee where there is no bee on the screen. And again, this reminds us that every experience you have is some combination of the remembered past and the sensory present. The prediction signals in your brain and the sensory signals from the surfaces of your body. Physical signals are constantly streaming from those surfaces to your brain. On their own, these physical signals have no inherent psychological meaning. And what I mean by that is when, if you hear a loud bang, your brain is trapped in a dark, silent box called your skull. It's receiving signals which are the outcomes of some set of changes in the world, but your brain doesn't know what the changes are. It only knows the outcomes. So your brain has to solve an inverse problem. And that inverse problem, in terms of guessing causes from only knowing the outcomes, is exactly what your brain has to do with the body as well. So if you hear a loud bang, your brain doesn't know what caused that loud bang. It could be many things. It could be a gunshot if you live in the United States in certain places. It could be a door slamming. It could be thunder. Same thing with a tug in your, a tug in your chest. What is that tug? Your brain doesn't know, it has to guess. And those guesses create meaning. Those guesses are prediction signals which create meaning. So physical signals become meaningful in relation to the prediction signals that are already assembled in your brain. And we as an ad hoc category, and we had this experience, for example, with the blobby bee. We also had this experience, even in the absence of sensory signals, with the color red. So one way to think about this is that these sensory signals are obtaining psychological meaning in relation to the prediction signals that are in your brain. When incoming signals match the prediction signals, then those incoming signals are categorized and explained. When incoming signals don't match, we call that errors of prediction or prediction errors. And when your brain encounters prediction errors, it can adjust its predictions, and we have a fancy name for that. We call it learning. Learning is your brain's ability to take in prediction errors and incorporate them into memory, consolidate them into memory so that they can be used for future predictions. So this is a simplified explanation, but I think it communicates the main hypothesis that category construction is really meaning-making. Your brain starts with a category of possible futures, and it ends with a specific instance of action and mental features. And I want you to notice what I've done here. What I've done here is I've completely changed the definition, in a sense, of categorization. So we usually in cognitive science, we think about categorization as there are categories out there in the world, and an occurrence happens, we retrieve the concept, we compare the concept to the occurrence, and then we plop the occurrence into the category. But what a predictive processing account suggests is that, in fact, what's happening is that the brain is starting with a category or a concept, because, there is no distinction, and it's receiving sensory signals that it has to make sense of, and the result is an instance. There's so much more to say about that, but I don't want to run out of time, so I'll just let you ponder that and say that in psychology, most of the major topics are about meaning-making, how the brain makes meaning. And so in this sense, I think this predictive processing approach, with our particular spin, when we think about prediction signals as category construction, unites these different folk psychology concepts into a common neurobiological framework, with the common core being efficient energy regulation of the body. And so every action you take, every experience you have is always some combination of the remembered past and the sensory present. And in our lab, we think about this as a relational view of meaning. Now, I want to point out something very, very ironic, what I think is ironic, and I hope you will too. If you remember the brain system we talked about a moment ago, the one that regulates your body as it's controlling your actions and creating your thoughts and your feelings and your perceptions, I'm going to present this to you again in a different format. So what I'm going to do is I'm going to show you a map where what I've done is I've taken one cerebral cortex, which is like one hemisphere, is like a balloon. Okay, it's like a balloon that covers the subcortical areas, and I'm going to pop the balloon and I'm going to lay it flat. So that's what you're seeing here. And I've colored the allostatic system in yellow and red and orange, and the hole in the middle is where the subcortical areas would be. So who here has heard that their brain has a limbic system? Okay. the limbic, so we understand the limbic system to be this really, really reactive system for emotion that has to be controlled by cognitive means, by rationality. But the term limbic actually was coined first by Paul Broca in the 19th century. It's an anatomical designation. It means border. It means regions that border the subcortical regions. So that's literally what limbic means. And these are the areas that border the subcortex that are considered limbic. So the term started out as an anatomical, a structural label, but it took on a functional meaning when scientists decided that these regions were the most important regions for emotion, the most reactive parts of the brain that were in need of control. So this is the irony. More than 30 years of published research on non-human animals, track tracing, looking at the connections, the literal anatomical connections between cortical areas by Helen Barbas, the anatomist Helen Barbas and her colleagues suggest that limbic regions are among the most powerful predicting regions in the cortex. They are not reactive and in need of control. They are predictive and they are doing the controlling. So they're not for emotion. They are primarily the main cortical sites for controlling allostasis, and they are where prediction signals originate, and then those signals cascade out to the sensory edges of the cerebral cortex. So I want to, if we had more time, and I do have slides prepared for this, I could show you the anatomical gradient in the cerebral cortex that we're talking about here. This is an anatomically inspired hypothesis based on the cytoarchitectural connections between cells, between pyramidal cells mainly, in the upper layers of the cerebral cortex. But the point I really want to make here is I'm not saying that a category or a prediction signal exists only in these areas. The prediction signals start here, and they cascade out and become more particularized and decompressed until they reach the sensory edges of the cerebral cortex. There's also a decompression gradient from the cortex down to the spinal cord. So the prediction signals are the whole cascade, the whole brain event. They're not just these compressed signals that are in these limbic regions. So here's just a quick gist to show you the gist, but I could also show you more specifically. The idea, based on Helen's structural model of cortical signaling, is the following. The first volley of signals in category construction are the allostatic control signals from that limbic ring, which are decompressed along the anatomical gradient from the limbic areas through the midbrain to the brainstem down to the spinal cord, where they become specific signals for particular movements. And at the same time, copies, literal copies called efferent copies of these signals off collaterals from the same axons move along the cortical gradient as sensory prediction signals that become more and more and more particularized into specific sensory predictions, which anticipate your experience of yourself in the world. So the hypothesis is that your brain constructs your experiences, what you see, what you feel, what you hear, as it predictively controls your body. Prediction signals, you can think of them as momentary, situated categories, ad hoc categories that your brain constructs that give meaning to incoming sensory signals. So your brain is, in a sense, guessing at what the meaning of those signals might be before they arise. And then the ensemble of signals, the multimodal ensemble of signals, helps to select which prediction signals actually gain access to motor control. So this is our lab's version of predictive processing, and it's as interpreted through a constructivist lens. And as Georgia mentioned, constructivism in psychology, psychologists sometimes think of constructivism as anti-realism, as idealism, really. There's so much to say about that. But all I'll say is that in recent work, we've started to consider what these ideas mean for the nature of reality as relational realism, which is not idealism. It's not a form of anti-realism. It's not saying that everything exists in your head. It's saying that what is real exists in the relation between the sensory signals that come from the sensory surfaces of your body and what's in your head. Okay, so in the last few minutes, I'm gonna, for the FENS meeting, I was asked to say something about the importance of allostasis in development, and which I did, even though I am not a developmental neurobiologist, but there are some, I think, really important implications here, and so I'm gonna just say them now so to give us an opportunity to talk about what that might mean. And again, I'm gonna emphasize here that these are implications, which means this is an example of using this allostatic narrative as a generative tool for generating new hypotheses. Because in my view, you know what, I'm not going to say that. I'm just going to say this and then we'll finish up. So otherwise it will take us in a different direction. An infant brain is not a miniature adult brain. It's a brain that's waiting for wiring instructions. Those wiring instructions come from the infant's own body and also from the surrounding world, the actions and words and so on that the infant is exposed to. So in the first few months of life, for example, a baby's brain becomes wired to see with two eyes at the exact distance of those eyes in her skull. So if we could magically take a person's brain out of their skull and put it into somebody else's head, they wouldn't be able to see. Your ability to hear is wired specifically to the shape of your ear. And if something unfortunate happened to your ear, you would have to relearn how to hear. So an infant's brain wires itself to the infant's body, and that also includes the internal sensory signals from the body. Little brains have to develop the wiring to manage allostasis, right? A little brain can't walk herself to the fridge when she's hungry. She can't put on a sweater when she's cold. She can't tuck herself into bed when she's tired. Babies can't even burp by themselves when they're born. I mean, really. Am I right? I'm telling you, it's like, yeah. Every parent is thrilled when a baby finally figures out how to burp. It's like everything gets much easier, not completely easier, but a step easier when that happens. But a baby's caregiver have to manage her allostasis for her by feeding her, changing her, burping her, setting sleep times, cuddling her, speaking to her, acting towards her in a way that is, dictated by the culturally inherited norms and practices of where they live. And this also includes managing the uncertainty for infants so that, part of what we do for our children is we titrate how much uncertainty they have to deal with, because uncertainty is incredibly expensive for every brain, but particularly for little brains. So if a baby feels safe and attached to her caregivers, these are manifestations of the efficient social regulation of allostasis. The baby's brain is constantly receiving wiring instructions that will help her perform allostasis on her own. Her brain is going to wire itself to her world, whatever the instructions she's getting. If she's getting great instructions, that's what will wire her brain. If she's exposed to adversity or neglect, those are the wiring signals that are going to wire her brain. And in a paper a couple of years ago, we hypothesized that this social foundation of allostasis is also the foundation for most category and concept learning, including the learning of abstract concepts. So infancy is one crucial period of social support of allostasis, and so is adolescence. Adolescence you can think about as a perfect storm of metabolic burden, where the body becomes more difficult to regulate for all kinds of reasons, like these. And so this is, again, another time during adolescence, when we are the caretakers of each other's nervous systems. We're social animals. We regulate each other's nervous systems. It actually doesn't matter what you believe, politically or morally. This is just in the wiring of how humans work. There's tremendous amount of evidence that this is the case. We don't understand exactly all the details, but we do understand that humans are social animals. We evolved as social animals, and we regulate each other's nervous systems for better or for worse, right? The best thing for a human nervous system is another human. The worst thing for a human nervous system is another human. But this sort of the difficulty of the brain's difficulty in regulating the body during adolescence is important. I think it's an important insight because it helps us sort of look at the conventional story of brain development and the affective upheavals of adolescence in a new light. So traditionally, the story of brain development is rooted in the myth of the triune brain. This is the idea that the human brain is born containing an ancient inner beast with circuits for instincts, your so-called lizard brain, and for emotions, your so-called limbic system. And that brain development involves the slow development of the cerebral cortex to increase its control over this inner beast until you finally become a rational adult around the age of 25. Well, we've already established the fact that you don't have a limbic system. You certainly have limbic areas that are, limbic by anatomy, but they're distributed across multiple networks. And you also don't have a lizard brain. And you know how I know you don't have a lizard brain? Because mammals didn't evolve from reptiles. Both mammals and reptiles evolved from fish. So this narrative that infants and children and moody adults supposedly have an underdeveloped cognitive system that can't keep hyperactive emotion, the emotion system in check, is not supported by the available evidence, despite the fact that it is the dominant view of brain development and of mood disorders. It's the dominant explanation of mood disorders, hyperactive emotion circuits that are not controlled properly by cognitive systems. I see you're looking at your watch. I'm wrapping up. It's just hard to give up a pulpit. You know what I mean? Like, I'm looking. Oh, I thought you looked at your watch. So. Oh, okay. All right. So I think that the narrative that I've just provided to you, grounded in allostasis and predictive processing, is a more justified interpretation of the existing findings, and it generates really different questions about what might be happening in development and what might be happening in mood disorders, particularly as they occur in adolescence or in illnesses or in menopause and so on. So to summarize the implication here, I'm saying brain development is not the triumph of cognition over innate emotion circuits. And really, we should stop saying that. Instead, brain development involves the necessary wiring for efficient allostasis and energy management, which supports flexible category learning and category construction, all of which play a role in mood and cognition and behavior, and more importantly, or more generally, I would say, in the construction of a human mind. Thank you. Absolutely, absolutely.
So I start because I'm in the fact of two questions if I can. Yeah, so Lisa, I think we discussed a little bit this, and my first question for you is starting from your last point, and how we use social or caregivers to regulate our nervous system. So my question for you is that most of the literature and also the sheer works is talking about what happened after birth. But we have colleagues here and also literature around that is more and more looking into the womb. So before even the child is exposed to the external world, but is, let's say, in touch with the internal mother body. So the question for you is when we start to think about the brain as an allostatic machine and a predictive machine, whether we can already do it when the child is not yet in the world.
So we've already had a very brief discussion about this. I'll just backtrack and start as if we haven't had that discussion for the benefit of everybody else. So I think at the outset what I want to say here is I want to be very careful because there are very serious political implications about what I'm about to say. So I'm just setting those aside. I'll just answer you like a scientist to the best of my ability. I don't think we know when, I don't think it's known really when, well, let me say it this way. Neurons are set up into a nervous system in an embryo. When do those neurons start functioning like a brain? I don't think people know the answer to that question, but I think we do know some things. I think we know in the third trimester, which is when in humans, when primates, when epigenetics are most, that's epigenetic influences are most strong, the brain is functional. Because fetuses there's evidence that fetuses are interoceptively sensitive to the mother's, some of the mother's interoceptive signals. For example, we know, I think there's very good evidence from Michael Meany and other people's laboratories showing that some of the mother's hormonal responses, specifically her cortisol responses, condition the nervous system of the gestating fetus. The only way that can happen is if the fetus is detecting those signals, right? I think one big question is really how much of the hormonal signaling the fetus has access to of the mother's signaling. But there's also there are these really intriguing other sources of evidence. Like if you take a premature infant who's been born prematurely after 6 months, and you in the in the intensive care little basket that you have the infant in, you play an audio tape of the mother's heartbeat, the way it would sound if you were in a uterus, it actually stimulates the development of auditory cortex in the infant. So that suggests that the fetus is tracking actually some of the interoceptive signals of the mother. Some of the hormonal signals also, though, maybe electrically some signals. The question is how much? And I don't think we know the answer to that question. But I think it's important to understand that some of the things that we think of as innate might be epigenetically wired. And I think, we run this, Karen Quigley and I there's the co-director with me of the lab, we run this interoception work group for the National Institutes of Health, for the National Cancer Institute specifically. And we got very excited. There was a big multi-site imaging study that was about to begin that was going to start imaging pregnant mothers with their fetuses. And we got very excited by everyone actually got very excited about the possibility of basically using the peripheral physiological signals from the mother and looking for shared information with bold signaling in the fetus. So everyone was super excited about this. The program officers were super excited about this. Everyone was excited about this, except it never happened. And you could imagine why it never happened, right? In part, it didn't happen for pragmatic reasons, because everybody wants a piece of the study. There's a lot of things imaging to do, could they fit it in, But there are serious implications about what you're asking. And so I think it's worth, we're not going to have that discussion here, but I think it's worth talking about those implications as we talk about, the hypotheses and what we want to learn and what implications they're going to have for, how that information might be used, I guess. But I think there's no question that part of the infant's temperament, what we call temperament, right? But part of the development of the nervous system. So all the neurons are already there. All the neurons that are ever going to be there, other than in the hippocampus, which in humans, that you can, the hippocampus can still birth new neurons after birth, can generate new neurons. It's really the only part of a human brain that I know of where it's clear that there are new neurons being born all the time. Everywhere else, when an infant's born, it has all the neurons it's ever going to have. But there's significant tuning and pruning that happens in the neural connections, and there's significant changes in myelination and so on. And there are significant changes in the metabolic efficiency of a developing brain. And all of those are, I think, fair game to ask questions in terms of the question being how much of the mother's interoceptive signaling is the infant, the fetus, actually privy to. I suspect it's more than we think. Yep. Oh, yeah, I just have something else, but...
Should I? Was someone else first?
I just thought of something else but I don't know if I want... I will just say one more thing. This is being taped. Is this being taped?
Yes.
Okay, maybe I won't say it. I'll say it later. Just come up and ask me. Yeah, go ahead. Let's ask a question. Yeah.
Thank you for a talk and beautiful slides. I was just wondering, where does the need for category comes in? Because you said the main job of a brain is to kind of regulate the body. And if we're thinking about the body, we're thinking about differences. We're thinking about continuous variables, like my heart rate going up or down, but it's not oh, I have a good heartbeat or a bad heartbeat. So why the need for category? Or should I think of category as not categorical variable and something completely else?
Yeah, I'm not thinking about a category as not thinking about a category as a categorical variable. So I'm sort of readjusting to a very generic definition of category, which most cognitive scientists I don't think would argue with, which is a category is a group of instances which are similar in some way. They're similar enough to be treated as equivalent. And they're not you don't have to, you can have categories that don't have firm boundaries, that don't actually have punctate boundaries. So the issue, it's actually, honestly, it's an anatomical hypothesis. I did not plant this question, but it does give me the opportunity to show you. To show you this, which I'm going to show you again, but in a different way, okay? You've seen this before, but I'm going to show you differently. I hope this will answer your question, okay? So, there is an anatomical gradient in the cerebral cortex. And here I'm primarily referring to pyramidal neurons, and I'm primarily referring to the upper layers of the cortex, okay? Here's the gradient. The gradient is, at one end you have very large pyramidal neurons, which have many, many, many connections. And there are not that many neurons compared the sensory edge, which have a lot of tiny little pyramidal neurons, and they have very few connections. So if you think about it, the receptive fields at this end are things like tones and edges and lines and really, really small particular details. And the receptive fields, so-called, at this edge, this limbic edge, are multimodal compressed summaries. So this is partly where the debate comes in about whether we have embodied cognition or not. Because some people want to say, this is amodal, these are modal, this is amodal. But they're not, these aren't amodal. There's actually, in the Shannon sense, sensory signals. So that these signals carry some information about sensory signals. So these are more like compressed multimodal summaries. This is what we would call an abstraction as a multimodal summary compressed. Okay? So imagine you go from here to here. So let me back up. Imagine you have prediction error coming in. What's happening? The signals are being compressed, like an MP3. Okay? I mean, basically like an MP3. Well, I mean, that's just an analogy. Okay, it's not but what you have is you have lossy compression. So you're getting rid of redundancies. You're reducing the dimensionality of the signals. You just get here. We have a multimodal compressed summary. Okay, this is where prediction signals start. Hippocampus would all be here too, but it just isn't because we made this fast. So when you go from here to here, it's an inference because you had lossy compression. But what happens is, at one stage, this is, let's say, an abstract feature, and then as you move towards the sensory surfaces, you have more and more and more options for peripheral, sensory and also motor particulars. So a bunch of different sensory motor patterns are treated as similar. They're considered similar in terms of the abstract features. That is a category. These are features of equivalence, and the instances that are possible instances are the sensory motor particulars. So I literally mean a category, but I don't mean that there's this, I'm not even talking about, there is a prototype for the category in an instance, because these are ad hoc categories. This is exactly how Larry Barcelo talks about ad hoc categories. That in the moment, the brain is it's making predictions about the possible different sensory motor particulars that have a similarity, a commonality, in terms of the abstract mental features, the multimodal compressed features. So threat can be any number of ensembles of sensory motor particulars. And if you only use the abstract features as features of equivalence, how large would your category be? It'd be huge. It would be huge. That's not very metabolically efficient. That's overgeneralization. On the other hand, if you only use if you didn't abstract, if your brain wasn't set up to abstract, that would mean that your features of equivalence would be concrete sensory motor particulars. So you wouldn't be able to generalize very much. Also not very metabolically efficient because you're trying to pattern match an instance to an instance. What kind of brains do that? Neurodiverse brains. Neurodiverse brains tend towards pattern matching and they have difficulty with abstraction. What kind of brains overgeneralize and have these massive, like very, very, the granularity of the categories is very low because they're using abstract features like threat or reward, or red, or red would be somewhere here, but overgeneralization. There are some mood disorders that function like that, or could be described that way. So I do think that category is the right way to think about it, but I'm not talking about firm boundaries, and I'm suggesting, as Larry does, that the prototype of the category is the ideal instance of the category in that situation. Right? The category is momentary and it's situated. So these are functional, these abstractions are functional abstract. And I want to point out that what the anatomy looks like, what it looks like, is that these regions are creating multimodal, they're reinstantiating multimodal summaries that are decompressed into allostatic signals, visceromotor and skeletal-motor signals, as they move down, from the through the midbrain to the brainstem to the spinal cord, where things get very specific. Actually, beyond the spinal cord, where they get very specific for the peripheral nervous system. That's a decompression, and then there's this other decompression. Now, I just say one other thing about this, and that is that there are decompression gradients there are decompression gradients, but compression gradients in the cerebellum and also in the hippocampus. The hippocampus itself is considered to be, from the perspective of the subcortical regions to which it is densely connected, and also from the perspective of the cortex to which it is densely connected, it's considered to be it's compressing signals in both directions. So the definition that I'm using of a category has very much to do with the anatomical compression gradients in the brain. And you can think of at every inferential step, at every synapse, essentially, you've got more compressed features being decompressed into, you've got a compressed feature being decompressed into more sensory, some things that are more particular, and more particular, and more particular, and more particular. So that's the sense in which I mean a category, if that makes sense. But I wouldn't interpret it as firm boundaries with like categorical distinctions in that way, if that makes sense. Yeah. Yes. Sorry.
Firstly, thank you so much for this amazing talk. I think this was one of the most fun academic talks that I've ever seen. That was really informative and fun. It was great. I wanted to go back to sort of the middle of your talk, where you said, well, the brain's job is not thinking and the brain's job is not seeing. And then you go ahead and...
I would say it's not its job. I would say it's not its main job.
It's not its...
important job. It's most important job. I think I'm very careful with the language here. It's most important job.
It is. So yeah, I wanted to talk about the language, because then you go on to say that the main things that a brain does is predictive processing, category construction, and learning as a way of error management. And as far as I know, I'm in a cognitive science lab as well. That is what thinking is, and that is actually what seeing is as well, because we are adhering with the Bayesian brain model all the time. And
well, I'm not saying that you should throw out thinking. I just don't think thinking is not a process. Seeing is not a process. I would say it's a result. It's an outcome.
okay.
It's not a process, right? So there's a thing that psychologists do. I probably did too for a long time, which is you say, oh, okay, there's a novelty. Novelty is a feature, so there must be a process for novelty. So there's novelty evaluation, right? Where you feel unpleasant, or you feel negative, or you perceive a threat. So you perceive a feature which is threat, and therefore there must be a threat detection mechanism in the brain. There's a parallelism between the products and the mechanism. And I'm just saying, break that. Go ahead. Let's think of, if you want to define thinking that way, be my guest.
I mean,
but
yeah.
I don't think thinking is a process. Thinking is a result. It's an outcome.
Okay, I don't think thinking is a result. I think thinking is a process, and seeing is a process, because if you adhere to the Bayesian model of the brain, then seeing is a process.
Well, I would say prediction is a process, and I mean, maybe we're just splitting hairs here, but I don't think so. So I would say, maybe I'm making a philosophical, maybe I'm concerned about something philosophical that doesn't concern you.
Yeah.
But, who here has heard sort of David Marr's 3 levels. Okay, so you've got, right? I'm saying, let's break the parallelism between those 3 levels. That's what I'm saying. Do you want to talk about seeing at the computational level? I'm fine with that. If you want to talk about seeing as a process, I'm less fine with that. And if you want to talk about seeing as localized in visual cortex, I am very not on board with that.
I'm not saying that at all. Yeah, not
so
saying that at all.
It matters what you call a process. And I think that's my response. Yeah.
Okay.
So thank you very much. So it's very good to see that your talk doesn't get old and not boring at all. So maybe not. It's like, you know, binge watching your favorite TV show over and over again. So it's very good that we still have this slide up, because an interesting point that I observed. So you talk about expansion gradients in the anatomical sense. So you mostly focus on the pallial expansion gradient, but there's also the subpallium with the olfactory system and the amygdala in the center. And you haven't talked about the amygdala at all. And people know me here that I love the amygdala. So what's your current hypothesis of what the amygdala is doing in your particular network?
What my answer will be in a few months from now. Let me back up one step and say, I was just going to put this up while we were talking so people could take notes or whatever. Let me just get that up there. Okay, great, okay. So here's this is something I've struggled with for a long time. And not just me, like I work with a bunch of engineers and computer scientists and other neuroscientists, and we have this group that meets like every week. We've been meeting every week for like 10 years. And one thing we struggled with for a really long time is there one model? Is the brain have one model, or are there different models? Meaning that the cortex, the cerebral cortex, and the hippocampus, but I would now include also the olfactory, the olfactory bulb. I would include the olfactory system in that now because of evolutionary considerations, and the cerebellum. Are they all integrating and compressing over signals differently? They're all getting the same signals, generally speaking, but they're doing something differently, or is there one model? So that's the perspective that I come at this from. I think the amygdala is really complicated because if you believe Larry Swanson's perspective, right, there's this beautiful paper by Goritza Petrovich and Larry Swanson in 1999 where they look at the amygdala, the different amygdalar subnuclei, and some are considered, striatal and some are considered cortical. So do you think of it as one structure, or do you think of it, right, exactly. So if it's the case that the cortex is, so the brain's a signal processor. I don't mean an information processor. I mean it's a signal processing, right? Okay. So, like this is not a 1980s view of like computer. I'm not saying the brain is a computer. I'm just talking like maybe more like engineering, I guess. But the brain is doing signal integrating. It's compressing, whatever. It's manipulating signals. If you think that the cortex is doing signal processing differently than the hippocampus, then you would have to say that the amygdala it's different subnuclei depending on their origin, their embryonic origin, that they're modeling differently, that they're part of doing models, which is what I think. So I would say the central nucleus and the striatal parts, I think, are really part of mainly allostatic regulation. And I think the nuclei have more to do with uncertainty, actually. With the precision of, I think they're involved in setting the precision of predictions and prediction errors. If that's it. So an old way to say it would be novelty, or uncertainty, or, right? So the way that maybe 10, 20 years ago, Paul Whelan would have said, the amygdala's function is to signal to the rest of the brain when there isn't a good prediction. And the brain has to update, has to learn in order to predict better next time. Really that only, I think that fits better with the cortical, just like the basolateral nuclei, things like that. But I'm a little uneasy about that, and the reason why is that I think learning is hard. It actually requires metabolic resources, so there's probably crosstalk between the nuclei in an important way. I also think that part of what the brain is doing, what salience is the brain is predicting it's making predictions about the prediction errors that are likely to be allostatically relevant. So what lack of salience means is this is not going to matter. I should not expend the energy learning this because it's not going to matter too much later. You would only engage in the expense of learning if it was likely to prove to be useful later, or if you didn't know and you were foraging, like you just weren't sure, right? Does that make sense?
Yeah.
So that's kind of how I'm thinking about it. The thing that gives me pause and why I might not, if you ask me in 6 months, I might not say the same thing, is that the way compression is happening is very different in those two systems. It's very different. So compression in the cortex is all about, there's spatial information that's maintained. Like it's spatial and temporal compression. So V1, A1, S1, these are all what you would call array-to-array maps, meaning the cortex is mapping the array of the sensory surface. V1 is a map of your retina. So nearest neighbor cells in the retina are nearest neighbor receptive fields in the cortex. But olfaction doesn't work like that. Olfaction is all the spatial information pretty much is compressed away at the nasal epithelium. The only way you get spatial location out of the olfactory system is if you move your head. So animals that can move their heads can get that information. But all of that spatial information is gone. It's only temporal contingencies, right? And for the interoceptive system, Karen and I, with one of our graduate students, just wrote a paper about this also, just summarizing the most recent molecular genetics work on the vagus and the nodos ganglion, which is the ganglion that is like where the cell bars are for the vagus. And it looks like, I want to say, it looks more like the olfactory system than it does like vision. Okay, there is some space, there is no visceromotor map in the vagus. If there's a visceromotor map anywhere, like a map of organs, anywhere in the brain is computed because it's not in the vagus. There's like a general kind of like top-bottom, is it thoracic or is it your gut? And there's like there are two sort of embryonic polarities in the way that the signals are being compressed. And there's temporal information, but that's it. So it's sort of more like olfaction. So that's why I'm saying I don't know, because I don't know enough about the amygdala's relationship to the olfactory system to really know, to make a good, well, really know, who really knows everything, but to make a good anatomical driven hypothesis more than what I've told you, but I suspect once we look a little closer, maybe there'll be something, maybe we'll learn something from the available anatomical work that will give us a clue. But what do you think?
Oh, I have a complex idea about it. So, but what I really liked about your answer here is that it's a different way of modeling, basically. So hippocampus to be more spatial based, and then amygdala more about temporal contingencies. And you also said that you change something if you act on the world, or if you turn your head and then you will smell different things, for example. So this action-orientedness, this is something what most of the fence people are crazy about, you know, about the amygdala outputs. And so this is, so what in a nutshell, I would say, I think it's the very important function of the amygdala is to link interoceptive modeling from the insula with extraceptive cues.
But I think that you could say that about every part of the, I mean, I think I could say that about the hippocampus. I could say that about, right? So, but how? Link how, right? So that's, I think, the key, right? Because you could also say that about, I mean, there are lots of places, you could say that about the colliculus. In fact, we do say that about the superior colliculus, that their sensory and motor signals, visceromotor and skeletal motor signals are the upper layer of the superior colliculus is actually visual. The rest of it is multimodal. So I don't disagree with you, but I think there are lots of places where interoceptive and exteroceptive signals are meeting, and also motor signals are there. But I think it's the way, maybe the weighting is different or something.
Yes, I totally agree. So, and I mean, you also talk about degeneracy, for example. Yes. And I think this is here in this context very important, because when you think of fear conditioning, for example, we see that the amygdala is involved and the hippocampus is involved to a certain degree. Yeah, for sure. And they might have different ways of doing it. Yeah.
If you look at the old literature, what they would say is that the amygdala is important for the primary learning, the primary theory learning, and the hippocampus is for contextual learning. But I think that's kind of missing. That, you know, that's like demoting. That's a very stimulus-driven. You know, I as the experimenter care about this, you know, signal, so I'm going to call it the stimulus, and I don't really give a shit about the rest of it, so I'm just going to call it context, and I'm going to say the context kind of modulates the processing of the stimulus. But I don't think brains work like that exactly. I think that brains are multimodal processors, and they certainly will weight certain features more than others, for sure. But that isn't necessarily the ones that you particularly care about as an experimenter. And I'll give you an example. SM, the famous patient SM, it is not true that she cannot do fear learning, if that's what you want to call it. She absolutely does fear learning. She goes to the dentist, it's really painful, she never wants to go back there again. Is that not fear learning? That's exactly the definition of fear learning. But she can't learn to mild aversive signals. So you can shock her electrically. She's not going to learn. It has to be really a whopping, the salience for her has to be, to what a neurotypical human would be, a massively salient signal, meaning it has massive implications for your future allostatic, predictions. That's what's required for her. Whereas for a neurotypical human with a properly functioning amygdala, it can be just a slight likelihood, right? Or on the other end, you take a kid who's been neglected, like a kid who's been exposed to, whose parents or caregivers haven't properly limited uncertainty for them, right? So they're exposed to neglect, essentially. Little kids, their amygdala starts to take on adult-like functioning. So everything is potentially salient. They haven't learned, right? So for them, for what a neurotypical human, we would disregard certain features as not being important enough or relevant enough. We could predict that's not going to be likely important in the future, so I'm going to ignore it as noise. For a kid who's been neglected and their brain has been wired, they're gonna, everything's going to be salient for them, which is vigilant. So that's the kind of thing I would say.
Okay, thank you very much. I think I have to hand over the mic now.
But we don't have to leave any, we have time here, and we're very happy that we could
Good. And at the very beginning already? Yeah. Okay, well, please go ahead then. Yeah.
Hello, thank you. I already read your book like 3 years ago, and I'm really excited to see you in person now because I think you're doing really great work. But what I wonder about, like the way you say it, and you repeated it several times, like the brain regulates the body. It seems really like a kind of one-way street, and I wonder, like, I mean, I don't know if I think about systems like the cardiac and the breathing system, they're kind of like interrelated system, and they have crosstalk, we have constant feedback loops. So I wonder if it also could be like, what role does like the other way play, right? How much role does the body play in regulating also the brain, basically?
Right. Well, I think I said that earlier, that in fact you can think about it the other way. And I said that several times also. But let me say this more generally. You decide as the scientist what the system is. Where to put the boundaries of the system, what's an input to the system, what's an output to the system. That's systems theory. In systems theory, you decide. You decide. So you could model the brain and the body as a single system. We could model the two of us having a conversation right now as a dyad, as a system. Or we could model all of us together in this room as a system. But one of the things that we have been concerned about is action. So if you assume that sensory processing is in the service of movement, motor movement, metabolic expense, you would talk about the brain regulating the body. But if you wanted to talk about, like I said before, you can talk about the signals of the brain constraining and giving meaning to the signals from the surfaces of the body. But you could also talk about the signals from the surfaces of the body constraining the brain's trajectory, its associative trajectory. So if you imagine a brain as a network that is completely unconstrained by anything, its memory can go anywhere. If it can do conceptual information, it can remember anything. It's an information gaining system in the sense that it can combine bits and pieces of the past and associate to all kinds of things which don't exist. And we have guardrails on that system, right? So the brain has its own guardrails. That's the frontal parietal control network. That's a guardrail. The salience network, that's a guardrail. The signals from the body, those are also guardrails. When we go to sleep, this is how I think of it. When we go to sleep, some of those guardrails are relaxed. That's why you can dream things that would never really happen in real life. Like, I don't know about you, but I fly faster on my back. You know, because my cape's not moving. You know what I mean? But have I ever actually flown in my life? Well, not really. Not like a superhero. But I've flown in planes, and I've, been on a hand glider, and I've watched, superheroes fly, and I've watched birds fly, and so on and so forth. So my brain can associate the things, it can remember things in the sense it can imagine things. Imagine, remember, it's all the same. It can reinstate, it can implement signal ensembles that would never actually occur in real life. But even when you sleep, your brain is attached to your body. And your body's signals, the interoceptive signals, are guardrails for the brain, not to go to certain places. And so I think, for example, when you take ketamine or... Sorry, what? Oh, yeah. When you take ketamine or you take psilocybin, what are you doing? You're relaxing the body's guardrails on the brain, which is why you need a guide. You need some kind of guardrail, and because of the brain's capacity to do conceptual combinations. So one way to think about this, which I mentioned earlier, is that the signals from the body are constraining the brain. They're constraining what the brain's predictions can be. And it may be that, for example, in depression, that constraint is the signals from the body have the brain locked down to certain predictions and not others. That's one hypothesis that we have. So there's not one right way or wrong way to think about it. There's a way that's more or less useful or more or less justified for answering a particular question. Do you understand what I'm saying?
Yeah, and would you say that predictions can also be generated outside the brain?
I don't know what that means. I mean, here's what I think. I don't know what it means to say predictions can be generated outside a brain. Here's what I know. I know that a brain can act on the world in a way to try to control the sensory signals that it receives. That's control theory. That's the sort of the flip side of a optimal control theory way of thinking about brain function is the flip side of predictive processing. It's taking exactly the same variables and just emphasizing some over the other, but there's not. But I don't know what you mean by that. That would be the only way that I could think of prediction. It's not really prediction actually, but what do you mean prediction outside the brain? Like
any other organ could have the capacity to generate predictions in the way that the brain does.
You have to have a memory to generate a prediction. And the only thing outside the brain that has a memory, sort of, is your immune system. Where do you think muscles have memory? Where does muscle memory exist? It exists in your brain, actually. Muscle memory is in your brain. That's what muscle memory is. Your muscles can't move on their own. Your gut can move on its own. Your heart, for a very brief amount of time, can move on its own. Skeletal motor muscles don't move on their own. They have to be stimulated by they need electrical stimulation to move. So muscle memory is in your brain. Just in the same way we say that we see with our eyes, but we don't see in our eyes don't see. You see in your brain. You need your eyes to see, but the seeing is in your brain. People talk, I don't know if it's popular here, but in the United States, it's very popular to talk about, trauma lives in your body, the body keeps the score. The body doesn't keep the score. To keep a score, you have to have a memory. And it's debatable whether you could talk about the immune system as having a memory. I think that's metaphorical, but okay, I'd be willing to go with that. But the brain keeps the score. The body is the scorecard. So there's no question that there is an effect of prolonged adversity on the body, but it is mediated through the brain, which is why you have to be careful. You can't talk about trauma as if it's in the world. Trauma it's relationally real. It's real, but it's relationally real because potentially really adverse things can happen to people, and they have no evidence of having a traumatic response to it because for them it wasn't traumatic. And there are other things where the mythical average person wouldn't experience something as traumatic, but some people do, and that trauma is real for them. That is real, not for them, but it's real. It's real. It's relationally real. So I don't think that you have to think about the body's control of the brain as being predictive. I definitely think that the body signals can constrain the brain, but in order to predict, you have to have a memory.
But would you say that the immune system might have something like a memory, so could it in theory then also...
It doesn't have a memory like a brain. I mean, the immune, look, in vertebrates, we have what's called an adaptive immune system, which means it can learn. And certainly, like an antibody degeneracy actually was, I don't know if it was discovered, but it was certainly, the immune system was like I don't want to say prototype, but ideas about degeneracy were formed in studying the immune system because a given antibody can work on more than just the pathogen that it developed in response to. But I don't think that degeneracy is the same as conceptual combination. I don't think that I would say that. And I think these are really good questions to ask, though, because I think for me, I think they're really good questions because they force you to unmask your assumptions, right? So I would say, I could go with the immune system having a memory of sorts because it learns. But I don't think it works in the same way. But I could be wrong. But I don't know enough to say that I can't make predictions that it would, or hypotheses that it would work in the same way as a brain. I don't think anything in the body works that way. But show me something that show me something that would make me change my mind, and I will. Yeah. Okay. Cool. That's cool. Sure. I'm not. Who's moderating here?
So
okay.
I have just two really quick questions. Thanks for the talk. So the first question is, you talked about that brain is like a prediction like brain does a lot of prediction. But can there be a scenario where it does? There is a scenario of higher prediction, which could lead to stuff like anxiety. Like
I don't know what you mean, higher prediction.
So more prediction, like brain predicting or trying to predict the future more or in a higher level.
I don't know why you need a higher level to explain anxiety.
No, I mean,
I think you can explain anxiety with what we have, but tell me more.
No, basically the idea that brain trying to predict too much of the environment, leading to anxiety. Can something like this happen?
Maybe. I think he's talking about. So I definitely think that increases in arousal can happen for sure because of predictive dynamics. So that's, I think, very well established. But are you asking? I'm just not sure what you're asking. So let me ask differently. Let me ask a question. Are you saying that the scope of prediction, meaning, so this is.
So I'm basically trying to say in a scenario where the brain tries to predict too much of what will happen next.
But too much. What do you mean by too much? Do you mean in the moment?
So by
it's
too much,
trying to predict too many signals. Do you mean? The precision weights? This is what you were saying. I think the precision weights are too strong. And so it's dedicating itself to one prediction and it doesn't have a big enough category? Like, what is too much?
So by too much, I mean that if there is a scenario happening and brain is trying to predict all possible things which could happen to that scenario, leading to something like anxiety.
Okay, so that there are people, so that it's not that is not too much prediction. What that is not enough precision in the predictions. And yes, there are scientists who who hypothesize that when the brain can't weight certain predictions more heavily than others, it has to maintain too many action plans simultaneously, which is very expensive. So there's too much uncertainty, and the brain's going to attempt to learn, and that requires chemicals which will increase arousal, and your brain will make sense of it as anxiety. Yes.
Yeah, something in this line I wanted to ask.
Yeah.
So,
but I think there's another way in which your question I think is potentially meaningful, and I think can I just say this? Or? Okay. So there's another way in which your question is really meaningful, though, and I think this is really relevant to the present moment. Maybe just for me because I'm here and I'm not at home. I think that humans right now, the things we care about, our allostatic niche, the things that are affectively relevant for us, our niche, the niche of things we think we have to predict, is very big. It's way bigger than it used to be for people, right? When I wake up in the morning, I am reading the newspaper to my horror almost every single day. But what am I trying to predict? My actions in relation to many, many things of which I have no control over. Like what's happening in the Ukraine, what's happening in Gaza, what's happening in, all these things which I care very much about. What does it mean to care very much about? It means it's allostatically salient. So I actually think that that's why I was asking about, I couldn't tell what you meant by how much, because there's also this niche issue of how big is a niche? And if the niche is too small or too big, this is also a problem. And I think partly why humans right now, I don't know if it's the case here, but in the United States, everybody's talking about how exhausted they are because, their fight and flight circuits are in overdrive. It's about uncertainty, and they're trying to manage uncertainty about many, many, many things over which they have no control.
Yeah, so in a way, brain is trying to predict a lot of things, or in which it cannot control a lot of things.
Yeah, it's right. And when I say it's trying to predict a lot of things, I don't think that I can control climate change, but I have to I have to predict every time I figure out what I have to recycle or throw in the garbage. That's like, I'm only slightly being facetious here when I say this. I have to figure out, there are so many things in everyday life that are uncertain, what I need to do next. And that's what predictions are for, in a sense. They're for regulating action. Visceromotor action, which supports skeletal motor action. In a sense, that's one way to think about it. You can think about it as, other ways too, but one way to think about it is, for action planning, you have to be able to whittle things down. And if everything becomes something that's uncertain, this is very problematic metabolically. And when I say that, I don't mean, you're going to fall apart in the moment. I mean, these little metabolic taxes add up over time. They take a long time to add up, but they do, and they add up to metabolic illness, vulnerability to metabolic illnesses, including depression. Yeah, and anxiety. Mm-hmm.
Well, we know there are more questions, but allow me a brief interruption, because there might be some brains that already signal that there is a need for liquid, calories, or whatever. And this, of course, is not a big picture, it's not changing the world, but I have another duty before we solve this or slowly see people moving back into life. I'm not loud enough. Sorry, Thomas. I only have 2 hands. So I brought you a glass of water now.
Thank you.
I handed over a glass of water because I learned it's something very relevant. Yeah. Also, it has an effect only in 20 minutes. But we might still be here. So I have to hand over our little present, because otherwise it is not be seen by the audience. And a present handed over in front of an audience is kind of a surprising, non-predicted thing for you now, because you're expecting more questions and answers, which will happen later. However, it comes as something that is more appreciated when many people see it, because it shows the generosity of the institution that hands over hands over the present. So, just as a break. Just as a break, we can go on with the discussion, but those who feel that their bodies signal need for calories and water, stuff is over there. Slowly go there, don't disrupt the discussion, but yeah, please allow me to say that I very, very much appreciated the talk. I also have my question, but I keep them, because I read the stuff first and then I come back on it. And at the same time, it's amazing that so many people joined and that you're so interested. And some of the questions we have to see, because we represent cognitive sciences in general, that some of the questions are very specialized for some of the people in the audience. And I also appreciate your patience with detailed questions about predictive systems in the brain. But it's just general. So I hand over the microphone again. And thank you all.
Thank you, yeah. I will say we probably can go on for about 10 more minutes and then we have to wrap up. I'll just reiterate that thanks to people for their patience for these very, very detailed questions and my very, very detailed answers. Sometimes I get carried away in the fun of the details, so my apologies. Thanks. Is it, who's moderating? Yeah, somebody has a microphone? Okay, yeah.
Test.
So yeah, thank you so much for the talk. It was really, really interesting and reminded me of a sort of never-ending debate in philosophy of mind about whether humans are more emotionally driven or rationally driven. I'm just wondering, how do you see this question? Is this a false dichotomy? Are you more leaning towards one side or the other? Or
I think that when a question's been around for a couple of millennia, it's probably the wrong question. I don't think it's a useful question because there isn't an emotional system and there isn't a rational system in the brain. I don't think it's a useful question, personally. Yeah. Thank you. I'm trying. We only have, 7 minutes, so I'm trying to titrate myself.
You said right at the start of the talk that every psychological phenomenon is a whole brain event. And I was just wondering, because there's a sense in which that could be quite a trivial statement. Like, as I'm asking a question now, my whole brain is active. As I'm thinking about what I'm going to eat for dinner, my whole brain is active.
If you think it's a trivial question, then I'm delighted. I think that's amazing, because it means that people will stop trying to localize psychological phenomena to particular parts of the brain, or networks, or whatever.
Right, so you would be against the idea that things can be localized to particular areas of the brain?
I'm not against anything. I'm just against any claims that aren't consistent with the mountain of evidence that exists. So when you say anything localized, you mean anything psychological, or you mean anything biological? What do you mean localized? Like, what are you talking about? What kind of function are you talking about here?
Just for example, something like vision.
Well, where would you localize vision?
The visual pathway, is that not just like a clear pathway with distinguished areas that can be distinguished?
Certainly the optic nerve and the optic pathway are really important for vision. I don't know that I would localize vision there. V1 is really important for vision, but I don't know that I would say it might be necessary for most kinds of conscious vision. It's not necessary for blind sight. I don't know that I would say it's sufficient. So you have to be really clear what you mean by localize. There's a really, really interesting study I think you should go look at. It's by Gonzalez Castillo et al. In 2012. I believe it's in PNAS, but don't hold me to that. But I think that's where it is. And they did this really neat experiment where they had 4 subjects, and they gave somebody a really simple visual attention task, these people, right? And instead of giving them 100 trials, they gave them 400 trials. And instead of finding, if you just give somebody 100 trials, you get blobs of activity in exactly the places that you would expect. Localized activity in early visual cortex and in dorsolateral prefrontal cortex. If you give people 400 trials, you get 70-something% of the voxels in the brain show a change in activity. And as a general rule, imaging studies, including many of my own from the past, undersample. They are not powerful. They are limited in their power in testing individual subjects. So you don't need to give a given subject 50 trials. You need to give them 500 or 5,000 trials if you really want to see the extent of activity change. But then what they did on top of that is they, instead of using, let's just say that they modeled the hemodynamic changes in the brain, which are the origin of the Bold signal, they modeled them more precisely, and they recovered, I think, something like more than 90% of the voxels showed signal change in the brain to a really simple visual attention task. I could go on and on and on, but basically, you pick any kind of study, not just an imaging study, but electrical stimulation study or an electrical recording study, you will always find evidence of information related to visual signaling elsewhere in the brain. And I'll just say one other thing. There's a study that was published in 2012, I want to say Keck et al., but I might be wrong, where it's sort of a horrific study, but it's an instructive study. So they took rats, and it might have been mice, I don't know, little rodents, who ran on a wheel. And they were recording from V1 neurons. And then they ablated the retinas of these rats, destroyed the retinas of these rats. And V1 quietened down, and after 24 hours, the neurons started firing in V1 again. Put the animal back on the wheel, the animal runs, same pattern of activity, or close. Well, where's the stimulation coming from? Like, what is stimulating those neurons? It's not the retina. 95% of the synapses in V1 don't come from the thalamus or the retina, like from the retina to the thalamus to V1. They come from other parts of the brain. And there's more to say about that, but what I'll say is it's really hard to claim that V1 is for vision, actually. It's possible, I mean, clearly it's processing signals from the retina, and clearly it's necessary for vision, but it's probably not sufficient. So you can take any part of the brain and think about it as what its primary purpose is and what it's an association region for. So, and from that perspective, the meaning of any brain region is in relation to whatever else you're in relation to the question you're asking or the signals that you care about. Even an axon, I took it down. But if you follow an axon, one axon goes many places. You could have a single axon from subgenual anterior cingulate that goes to a motor nucleus in the brainstem. So it's a motor signal. Collateral off that axon goes to a sensory region. So that's a sensory signal. So is the action potential a motor signal or a sensory signal? Well, the meaning of the action potential is not in the action potential itself. It's in the relationship between where the signal originates and where it arrives. The meaning of, right? So you can't even localize a sensory and a motor signal. So yeah, I think it just depends on what you mean by localize. Yeah.
Okay. Thank you. Thank you for the talk.
I think we're done. Yeah, I think we're done.
Well, yeah, I think we all thank again for this super discussion. It was like a lecture. I think we should invite you for a long, long series of lectures because the answers are so fascinating and based on the literature. So to all our students, don't stop reading. Also, it's so tempting in these days, but read the literature, know it, and then you get the arguments that are the answers to your questions, although sometimes the answers are very open and we don't know yet. But I thank you very much for this very differentiated answering to all our questions so far. And I hope that you enjoy a drink and something to eat. Thank you so much. So, wo tun wir das jetzt hin?
Das geht mit dem.
Oh! Wow, she's a great dresser. Thank you so much.